All right, welcome to Tosin Learns at Biology Clarified. This is 2026 WAEC Biology Practical Examination Review, Mastering the Practical for Excellent Performance. This is a complete specimen analysis, predicted questions, and examination guide. So let's go. Specimen list at a glance. You have specimen A, groundnut balls, crushed roasted groundnut seed, C. Beaker with 20 ml of water. Beaker with 20 ml of table salt solution. That's specimen D. Specimen E. Beaker filled with sandy soil. Specimen E is a portion of alimentary canal of small mammal from esophagus to the end of stomach. And specimen F is a twig or a branch of a plant with mistletoe. Next, we have specimen H, evaporated milk in a beaker, J, fresh fish, K, dry maize grain, L, tomato paste in a petri dish, M, fresh tomato fruit, N, rotten tomato fruit in a petri dish, P is thermometer, Q is meter rule, and the last specimen is R, quadrant. Well, the practical review objectives. The objective behind this practical review is to identify, for you to be able to identify all specimens, to be able to state the observable features of all the specimen, to show you how you can perform food tests, draw and label biological specimens correctly, and then answer likely why questions. This is just about exploring projected questions on all the specimens as requested by biology. Practical review benefits to candidates. As a candidate, what would this review do to you? It will prevent exam panic. Yes, because you will not be seeing the specimen for the first time. And you will have a measure of confidence that, okay, I've been exposed to a series of possible likely questions that will come out, and that will boost your confidence and you will not be panicking. Another benefit to you is that it fixes your rote learning blunder. What do you mean by rote learning? Candidates who simply memorize past questions, often for often fail when WAEC introduces a slightly twist into the question. And then with these projected questions, you are trained as a candidate or a student to think on on the go. Just think on your feet. Apply knowledge and to any specimen presented, and you will be you will be confident that in any way they tweak the question, you will still be able to respond. Another benefit is that it guarantees that your marks that is awarded to technical points is not being lost. It forces you as a student or as a candidate to practice strict exam compliance like sharp pencil lines, correct label, drawing to size, all of those things. You have the opportunity to practice them, knowing that you know the specimen specifically without crossing, or so you won't lose unnecessary marks. Then one of the last two benefits for you is that you'll be able to troubleshoot. Yeah, even on your own, you'll be able to if this is not what they're asking for, then okay, this is what it's going to be. If it's not A, then it will be B. If it's not B, then it will be like that. That's troubleshooting. Then it saves revision time. You're no longer guessing or anxious about what you are very much sure. Why? Because I am an examiner, I mark, and I understand the dynamics of how this question comes. That does not in any way say that we know what the question is. No, we don't. But we review it based on experience. So you have the specimen list overview here, pictorial representation of the specimen, granite pods, crushed roasted granite seeds, beaker with water, big cow with soil solution, big cow filled with sandy soil. This is a diagram of as the digestive tract of a small mammal from the stomach to the from the esophagus to the stomach. Then you have the parasitic plant here, Mistletoe. We have evaporated milk. So here you have the picture of fresh fish, dried maize grain, tomato paste, fresh tomato fruit, rotten tomato fruit. You have thermometer there, the ruler, and then you can see the quadrats. That's the picture representation of the specimen. Before you will go into the detailed conversation about the specimen, I decided to talk about WAEC examiners' expectations. You need observation skill. What do we mean? Students should observe specimen carefully and correctly observe color changes during food tests. Then your reporting skill. State only what you can see, meaning that you state only observable features of the specimen. Use biological terms. Write concise answers. Don't be writing stories. We do not mark stories in biology. We mark biological terms, points that are sensible, direct, no stories. Then your drawing skill, very important. A lot of candidates lose mark from drawing. Use sharpened pencil. I advise you use blade in getting the tip of your pencil very sharp. Learn a freehand sketch. Draw with your free hand. A free flow of drawing. Explore all biological drawing rules. Freehand sketch. Write your magnification title. Label your diagram. These are free marks. There are about 5 free marks there. The title of the diagram, the size of the diagram, you'll be given a specific size to work with. Make sure that your diagram is not lesser than the minimum size and is not more than the maximum size. That's 1 mark if you follow that instruction. You write your magnification. Your magnification is the diagram and the specimen, the original specimen, and the diagram, the fraction of it. What fraction of the original specimen did you draw? So you put it there. It does not have any measurements. It just times the number. If I'm drawing orange, for instance, how what's the fraction of the orange that I draw? Is it half of the original orange or is it times two of it? That's what magnification means. Then lastly, experimentation skills. These are the expectations you that is required of you in the exam or during practical biology. Ability to conduct simple laboratory experiments like food test, because food test is the major experiment that is going to be conducted in this 2026 WAEC biology practical. Correctly observe and report color changes. Write only what you see. Don't write what you assume. Don't assume what is going to be the color change. Write what you see only. Specimen A is groundnut pods. Groundnut pods, because it's going to be more than one groundnut pod that will be presented to you. The common name we all call groundnut pod is groundnut. The food type is nuts, is dry dehiscent fruit because it splits to open. Agricultural classification is legume or pod. It belongs to family Fabaceae. And then class Dicotyledonnae. Then the botanical name is always two names. Sometimes we call it scientific names. That means the generic name, which is the genus to which the organism belongs to, the specific name, which is the species to which the organism belongs to. So if we are looking at botanical name or scientific name across this video, I'm not going to repeat that again. The first name is Arachis, which is genus, the genus to which granule belong to, and that's generic name. And it always starts with capital letter. Why the specific name, which is Hypogaea, also All of them must be written in small letter. Very important. It is painful to know, it will be painful for you to know the spelling of a word. And this simple rule of using upper case and lower case will make you to lose your mark. So specimen A, groundnut pods, types of placentation found in specimen A with reason. Placentation talks about is about seed arrangements. So the placentation found in specimen A is marginal. Why is it marginal? Because the ovules are arranged in a single margin row, in a single row along the margin of an ovary. Remember when you were taught fruits, the ovary becomes the fruits and the oval becomes the seed. So in a granite pod, the seed that developed from the oval is arranged along the margin, single row on the margin. Then what's the mode of dispersal? The mode of dispersal is explosive mechanism. And then what are the nutrients or the class of food contained in granite pod? Protein, fat and oil, and carbohydrates. Question could come as state three observable features of specimen A. It could be two or three. No, at least minimum of four or five. Based on what you can see, it's the pod is brown. It has dry epicap. It contains seeds enclosed in a pod, consists of elongated shape. The constricted, there is a constriction between the seeds of the pod. Then economic importance, source of food for humans, used for edible oil production. You know your popular groundnut oil, used as like livestock feeds, is part of the content that they use in providing feed for poultry. Source of income from confectionery products, you know, your parfait, your kuli, the traditional one, egusi. We even use it as soup. That's a source of food. Here we see the drawing of the diagram of specimen A. So go and practice the diagram. You can pause this particular space to study it. All right. So here I decided to combine specimen B and specimen C, and you will soon find out the reason why I did that. So on the specimen B, you could be asked state two uses of processing specimen B, because specimen B is actually processed seed of specimen A. Extraction of refined cooking oil or edible oil extraction is also used as a source of food, as snacks, processed food, like for cooking soup, groundnut soup. They also use it as nutritional supplement for malnourished children. It's also a source of animal feed. It's also used for nutrient for improving soil fertility during crop rotation. Um, then let's go to the food test. You could be asked to take a portion of specimen B and smear it on a white plane, and it will give you a spot, a grease spot, which indicate the presence of fat and oil. So this is the reason why I combine specimen B and specimen C, because of food tests. I feel that specimen C is there, 20 ml water in a beaker. So you could be told to take a portion of specimen B and pour it into specimen C and stir it to get a granite solution. And then you will now be told to split them in portion to carry out the following test. And then I feel that the portion of the solution, the mixture of specimen B and C, will be used for protein tests, where you'll be asked to add sodium hydroxide. You will shake the mixture, then you add copper 2 sulfate, you shake it and observe the color change. From experiment, the color change was purple. You could write violet color. And anytime there is a color change in any food items that copper 2, the sodium hydroxide and copper 2 sulfate is added, it means that protein is present. If there is no color change, protein is absent. And if you are asked for the name of this particular experiment, it is known as Biuret tests for protein. The second experiment is use of Benedict solution. Anytime you see Benedict solution being provided, it is an attempt to test reducing sugar. Reducing sugar is a disaccharide under carbohydrates. So a portion of specimen B, a mixture of specimen B and C, if you add few drops of Benedict solution, it will have a brick red precipitate, and that shows that carbohydrates is there. That means there's a presence of sugar in that mixture. That means granules has reducing sugar. The brick red precipitates at the bottom. Then lastly, iodine. Anytime a food item is to be tested with iodine, know that they are looking out for starch. Test for starch is the use of iodine. And if you put a drop of iodine on carbohydrate, on anything that contains starch, it will turn blue black. But one thing that is significant about granules is that not all the crushed granule seed turns blue black. So you need to be very sensitive. Some parts retain the color of the iodine. Some part there is a trace of blue black change, meaning that granite seed contains carbohydrate in a traced amount. In a trace amount. That's it. That could be question asking for the food test used for identifying specimen B. So let's go. For identifying fat and oil content of specimen B, these are the translucent or grease spot test and Sudan 3 test. Then for identify the protein content of specimen B, Million's test, xanthoprotein test, and Barrett test. The one that was conducted earlier is Barrett test, the one that uses sodium hydroxide and copper 2 sulfate. Then food test used for identifying starch in specimen B is for identifying carbohydrate is iodine, which is called test for starch. And then for identifying reducing sugar, the test is called Benedict's test, where Benedict solution is used. Now, you could be asked to state two advantages of processing specimen A, the seed of specimen A into B. So, like trying to link the two of them. Easier digestion, easier extraction of oil, and easier storage use. Specimen E is sandy soil in a beaker. You could be asked to identify specimen E, state three observable features, two agricultural advantages, two agricultural disadvantages. Features of specimen A: coarse particles, large pore spaces, loose texture, light brown color. These are things you can see. Advantages of sandy soil to crop plants: good aeration for roots, easy root penetration, easy cultivation. Another point is disadvantages of sandy soil to crop plants. Low fertility, poor water retention causing wilting of plants. That means it makes the plants to lose water and dry out. Nutrient leaching or leaching of nutrients makes the plant to lose nutrient because the soil does not have the capacity to hold water, then it's consequently is unable to hold nutrient because it's water that will hold the nutrients. Then you have high soil temperature. Characteristics of sandy soil is another point that could be asked, another question that could be asked, and is similar to feature of sandy soil. Has large particle size, has large pore spaces, it is well aerated, has high aeration, has low water holding capacity, or you can say that it has poor water retention. Yeah. Then has low fertility and has good drainage. Just pick any five and know it, you know. So you could also be asked, how can a farmer improve the quality of sandy soil for agriculture? You basically the basic answer is adding organic matter or adding organic manure or compost. And why do you want to? Why is that answer right? Because organic matter binds the loose sand particles together. It introduces colloidal surfaces that hold nutrients and significantly increases the soil's water retaining capacity. Remember, One of the disadvantage of sandy soil is it has low water holding capacity, and it is water that will hold the nutrients. Another question could come as benefit of planting specimen A, that's groundnut pods, in specimen E, and your response can be ease of peg penetration, adequate aeration, or prevention of root rot. Yeah, unhindered pod expansion. When there is a growth of plant from seed to seedling, when it starts to produce fruit, the groundnut needs space to increase, to expand. So the loose arrangement of sandy soil offers minimal physical resilience, allowing buried pod to expand and mature without deformation. Another point is prevention of pod rotting because of the good drainage of sandy soil. So you could also be asked to state the three observable physical characteristics of sandy soil. Okay, it has a coarse, gritty texture when rubbed between fingers. See, I have different perspective of questions and responses that are similar. So this one is more, more. I would say you should go for this. It has a coarse, gritty texture when rubbed between fingers. You can go there and touch the granite sandy soil. It has large, loose, and non-aggregated soil particles. The particles they don't stick together. It is light brown. Some of them are grayish white in color. Whichever your teacher provide for you, that is the color you should stick to. Then it cannot be molded into a stable ball or ring when wet. You cannot, like, unlike the clay soil, that means granite sandy soil, sorry, crumbles easily. Now you could also be asked to differentiate between specimen B and specimen E. You remember what we are doing is postulating possible question that could come. We're not sure. So they could say, okay, compare specimen B, which is a crushed granite seeds, and specimen E, that is sandy soil. They are both, crushed, like graining. So I have brown color or light brown in color, that's specimen B, and then brown or yellowish brown in color for specimen E. Just look at the color difference and write. Powdery, that's for those who will mill their own. So finely milled texture. Then for sandy soil, it is coarse texture. You remember if they say observable features, that means you must mention feature structural. But if it is not feature, then you could say that granite oil, granite specimen B is edible, and sandy soil is not edible. You cannot eat it. Then the particle size is small and uniform, kind of, in specimen A, and specimen B, I mean. While for sandy soil, it is larger and irregular. So you can use food. One is a food, one is just a growth medium. You can use nutrients. One is a plant product, one has mineral material. So I decided to talk about the most likely procedure that will be required in this practical. The presence of filter paper that suggests that candidate may be required to prepare an extract from a small solid specimen. One, place specimen B, crushed granite in a beaker, add specimen C water, and stir thoroughly, filter using filter paper. They could say use the filtrate for protein test, biored test, reducing sugar, that's Benedict test, starch using iodine. I've said that here. So this means that WAEC may be targeting three food tests because the reagent that are asked to be provided is Bennett's solution. When Bennett's solution is provided, it is reducing sugar that you are testing. When iodine is asked to provide it is starch test for starch that you'll be conducting. And when sodium hydroxide and copper 2 sulfate is being requested, it is biuret test, and which is a test for protein. So this is the portion of specimen F where we are told to provide only the esophagus to the end of the stomach. I just give you the diagram of rabbits so that you can understand the organ before the esophagus, the organ next, and the organ after. You can pause this video to check to study the diagram. So if you are told to identify specimen F, is a portion of alimentary canal from esophagus to the end of stomach, and that it is from a small mammal. Organ present in specimen F is esophagus and stomach. Observable features of specimen F: muscular sac-like structure. Stomach is the structure that is muscular and sac-like. Then you have esophagus. So this stomach is connected to esophagus. Then we have cardiac sphincter and pyloric sphincter. I have some reservation for cardiac sphincter because you have to cut the portion that connects esophagus to stomach before you can actually view the cardiac sphincter. But it's there. Why the pyloric sphincter is for sure visible because it's the end of the stomach. So the duodenum is not going to be part of it. For teachers that are providing the specimen, don't do the mistake of adding duodenum in the course of cutting. Now, functions of observable features. Now, those features that you observe, the esophagus, the stomach, what are the function? Esophagus function is just to move, it's just the movement of food bolus, food from the mouth to the stomach. And the movement is called peristaltic movement. Then the functions of the stomach is a temporary storage of food. It's responsible for protein digestion. It's also responsible for secretion of gastric juice, which contain enzymes. And then enzymes, pepsin, renin is also acidic medium. Then it's responsible for churning and mixing food. Enzyme that acts in specimen F is pepsin and renin specifically. Pepsin converts protein to peptose or polypeptides. Renin causes or coagulates milk by converting soluble milk protein, that's carcinogen, into casein. Then insoluble, the casein is insoluble while the milk is soluble. Then you can have trace of lipase. Digestion of fat starts from the stomach actually, though digestion of fat does not take place in the stomach. But pepsin and renin require an acidic medium to function. Stomach contains hydrochloric acid, HCl, which provide acidic medium for pepsin and renin to function. And one of the things that the hydrochloric acid contained in the stomach does, it kills bacteria that is swallowed along with food. And it also helps to neutralize the alkaline saliva or the alkaline food that is coming from the mouth. So here we are looking at the adaptation of the stomach for digestion. It has thick muscular wall for churning. It has gastric gland for enzyme secretion, and its mucous lining secretion for protection. Then it's expandable. It has an expandable wall for food storage. I make a joke that those who are pot-bellied, they're pot-bellied because the stomach, that's a temporary storage for food, can expand. Thank you. And this is another specimen, the twig of plant with mistletoe. I must warn here that it is not mistletoe. It's a twig of plant. That means there are two plants involved. One plant is the host, while the mistletoe is the parasite. You can pause this slide to view and study the diagrams, the pictures. So we have likely question on specimen G. Identify specimen G without giving reason. State two observable features of specimen G with which indicate its mode of nutrition. What is the mode of nutrition of specimen G? Make a label drawing of specimen G. 6 to 8 centimeter. I just came up with that. It could be 8 to 10 centimeter depending on what WAEC gives you. Long to show features of biological importance. So these are the possible answers. Application of specimen G. Specimen G is a twig of a plant with mistletoe. What are the observable features of specimen G? It has green leaves or chlorophyll. presence of osteria or osterium. Osterium is a sucker or an attachment organ. It has leaves that are arranged in opposite form. It has branch stem, both the stem of the host and the stem of the parasite. Plant attached to the host and has flowers or has fruits too. So here I have mode of nutrition without reason. Parasitic or semi-parasitic or heterotrophic. Mistletoe is actually the parasite is the mistletoe that is exhibiting the parasitic nutrition. And autotrophic nutrition there because there are green leaves there. Their mode of nutrition with reason, parasitism. Parasitism because there is a parasite growing on a host plant and it depends on the host for all nutrients and causing harm to the host. Autotrophic or photosynthetic nutrition because it has chlorophyll. There is a green leaf, and every green leaf has chlorophyll, and chlorophyll is one of the conditions necessary for photosynthesis to occur. Adaptations of mistletoe, that's the parasite plant that is on the host plant. It has osteria or osterium for attachment to the host, for penetrating into the host body to absorb nutrients and water. It has fruit for efficiency dispersal. It has flower for high reproductive rate. What are the effects of mistletoe on the host? It's cause the host nutrient loss. It's cause a reduced growth on the host and it reduce yield for the host. So this particular slide is added for extension. You can take a look at the labeling on it. You can pause, take a look at the label, take a look at the functions of the label parts. The This one is the diagram of the twig that was extracted from the orange tree. You can take a pause. This is just an added value. I'm using this for my students. If you are told to state one benefit the parasite plants obtain from the host, water absorption, nutrients, and attachment to the host. So name two other examples of parasitic plants aside mistletoe. We have doda plants, that's cuscuta, meadows. Then it is very important to know that in parasitic plants, Wisteria are specialized or modified roots or stems that puncture or penetrate the host's outer layer or tissue to tap directly the transport of vascular bundles that is there. That's how they're able to take their nutrients and take their water from the host. So this is a specimen that has milk in a beaker, and then it's obtained from cow. Method of preservation of specimen H. Pasteurization, sterilization, dehydration. Dehydration means drying. And then method of storage. Don't go and be mistaking preservation with storage. So refrigeration, freezing, canning, or airtight packaging. So class of food present in milk, protein, you have lactose, and then you have that's milk sugar, and you have lipids, that's fat and oil. Those are the three classes of food contained in milk. The importance of milk in human diet, bodybuilding, replacement of worn out tissues, growth. Just go and read about the importance of protein and then fat and oil too. Then we look at tests. We also suspect that there may be tests under this specimen because of the fact that it contains protein and it contains lipids. So it could also be tested for fat and oil. But if it's whatever, what you should know with tests for food is that if sodium hydroxide and copper 2 sulfate is added to any food substance, if it turns to purple or violet, it contains protein. And if biuret solution is added to any item, and if it turns to a brick red precipitate, just know that reducing sugar is inside. Any color change different from that means that it is absent. Don't stress yourself, please. Specimen J is fresh fish. Remember, we were not given a particular species of fish to provide. Everybody has the freedom to go and look for any type of fresh fish. So this is my own choice. I chose tilapia. And then the habitat of fresh fish is freshwater habitats. Freshwater habitats. And I will advise that you can be as specific as possible, like saying river or streams. Don't go and be writing aquatic habitats. Nobody's going to give you the mark. It's too general. Then classification of specimen G, kingdom Animalia, phylum Chordata, class Ostaches. Please get the spelling. Try your best not to write Pisces. Ostachitis is the specific class of this fresh fish. Then observable reasons for, but you could write Pisces since it's a fresh fish, any fresh fish. So observable reason for classifying specimen J into that class because the body is covered in overlapping scales, presence of operculum, that's the gill cover, presence of paired and unpaired fins. The pectoral and the pelvic fins are paired, while the others are unpaired. They have the presence of lateral line. It is important to note that don't be pushed to write gills. You have to lift the operculum up before you can see gills. Thank you. So here we have the drawing of specimen J. I actually draw this specimen myself and took it to ChatGPT to enhance it. So that's how I arrive at this. So you can see the labeling, horizontal labeling, ruled, and the moment the ruled guideline is horizontal, the label is also horizontal. And I'm going to have mark for ruled guideline. I'm going to have mark for magnification. I'm going to have mark for diagram, title of the diagram, that's 3 marks. I'm going to have mark for size of the drawing, that's 4 marks. So clarity of line, because of what ChatGPT has done to the diagram, I don't think there's going to be any mark for clarity of line from what I'm saying here. But in what I drew, there's clarity of line. You cannot see the book here. I was with the book. All right, let's move quickly. Then you have observable reasons for class of specimen J. Body covered in overlapping scales, presence of operculum, presence of paired and unpaired fins, presence of lateral line, nutrients present in specimen J, protein, fat, and oil lipid, vitamins. Economic importance of specimen J, source of income, source of food or source of protein. Observing features of specimen J has streamlined body, presence of fins, presence of operculum, presence of overlapping scales, presence of lateral lines. You will see that there are repetitions of certain information because the dimension of the rest question still goes back to those features. Then you have adaptations to aquatic life. Now, streamline body shape. Anytime that adaptation is required, you are expected to mention the feature the organism has and the function of that feature. So, streamline body shape for easy movements or for swimming in water, has fins for movement and balance or fin for steering in water, gills for gaseous exchange, overlapping scales to reduce friction in water, lateral lines for detecting vibration and pressure changes in water, pectoral and pelvic fins, those are the paired fins for steering, balancing, and braking in water, caudal fin for forward propulsion, propulsion in through water. I will advise that only pick the pictures that you can see with your naked eye. Thank you. So we move to specimen K. We are told to provide dry maize grain. Agricultural classification is grain or cereal. Scientific classification, that's when you look at the class. The class of maize is monocotyledonea. The fruit type is caryopsis and is a dry indehiscent fruit. Is one-seeded fruit, is dry indehiscent, it does not split. So then the pericarp fruit wall is fused with the textile. That's the seed code. That's why it is called. That's the reason for the fruit. Class of food contained in specimen K is carbohydrates. Observable external feature of specimen K remains of style found at the broad, flattened upper tip of the grain. Then the remains of stalk visible at the narrow pointed base, indicating where it is attached to the cob. That point where it is attached to the cob, to the maize cob, that's the remains of stalk. And then embryo stroke germ groove is a slight depression or ridge on one flat face of the grain. The maize has a flat face, so that outlines the position of the embryo. That's where you will find it. Then the agricultural importance: pests that commonly attack specimen J, maize weevils and termites. Preservation methods: proper drying, storing in airtight containers, using pesticides or fumigants, heat treatment. Specimen K is a monocot, has one cotyledon. Leaves leaves have parallel venation, and they have what is called fibrous roots. You can pause each of the slides to study the contents. Here we have specimen L, M, and N. I combo the three of them because I believe there's going to be a relationship between the three of them in terms of question. Specimen L is tomato paste, specimen M is fresh tomato fruit, specimen N is rotten tomato fruits. Method used in producing specimen L from M: boiling, blending, straining, and thickening. The four methods are included in the response. Botanical name of specimen M is Solenum lycopersicum. Still on tomato paste, fresh tomato fruit, rotten tomato fruit. I think maybe they could say compare the three of them. So this is a table comparing the three. Fresh tomato firm, solid and succulent. Rotten tomato soft, mushy, watery and collapsed. Tomato paste thick, viscous and smooth paste. Then bright red color for fresh tomato, discolored with white, black, green patches for rotten tomato, then dull red color for tomato paste. Then for fresh tomato fruit, we have pleasant smell. For the rotten one, offensive foul smell. Then for the tomato paste, it's still a pleasant smell. Then when you look at the micro fresh tomato fruits is meant to have very insignificant amount of microbes in the external point, and then the rotten tomato is diseased, is spoiled, is highly infected with microorganism. While tomato paste is there's absence of microorganism if it is not yet spoiled. Then the fresh tomato we have high water content. In the rotten tomato you have moderate water content, and in the tomato paste you have dehydrated states. It is lacks water. The organism responsible for spoilage observed on specimen A is bacteria, fungi, mold. Then preservation of specimen M, or how can specimen N be prevented? There are two different questions, but the answer is going to be the same: refrigeration, drying, canning. Agricultural classification of specimen M is vegetable. Tomato is a vegetable. The type of fruit is a berry. And why is it a berry? It's a fleshy fruit that has many seeds embedded in the pulp. Types of placentation is exile. Placentation reason because the ovary is multi-ocular, meaning that it has multi-chambers, that's locus, and the ovules are attached to the central axis. Yeah, that's the reason why it is exile. Botanical classification of specimen F in detail: kingdom Plantae, division Angiospermatophyta, class Dicotyledoninae and Dicotyledonopsida. You have order Solanalis, Solanaceae, then you have genus Solanum, species Lycopersicum. So if you are going to call the botanical name in specific of tomato, you write Solanum Lycopersicum. Then agent of disperser is human, man, animal. So we have specimen P, QR, thermometer, meter rule quadrats. So specimen P is thermometer, Q is meter rule, Y, R is quadrat. These are questions you can pause to look into it. Study specimen P, Q, and R answers the following question. Name one branch of biology which involves the use of specimen P, Q, R is obviously ecology. State one function of each specimen P, Q, R, and R. Describe briefly how each of specimen P, Q, R, and R could be used. Draw a specimen QR, I thought. Name two organisms each associated with the use of specimen R. I feel that they will ask you to draw specimen R. So specimen P, thermometer, is used to measure the temperature of the environment, air, water, or soil. Temperature is the degree of hotness or coldness of an object, and the SIU is degree Celsius. Specimen Q is meter rule. It's used to measure distance, depth of water, length of an object, or an habitat. The common question could be state two uses of a meter rule in biological field. Answer: it is used to measure the height of herbaceous plants or grasses. It is used to measure the distance between sampling points, e.g., in belt transit sampling method, or the depth of a water body. Specimen R, quadrat. Quadrat is is used in sampling method, random sampling in ecology. The principle on which the use of quadrat for determining population are basis was a past question that I got from WAEC past question. The quadrat is used for stationary organisms, and at least there must be 10 throws must be made, and then the size of the quadrat depends on the organism being sampled. Still on specimen R, quadrats. Function of specimen R used to estimate the population of plants or determine the population of small organisms, small plants, or crawling insects or population studies. Anyone, anyone. The organism associated with the use of specimen R is quadrat. Ants, millipede, beetle, garden snail, triders, water leaf, or talinum. That's the scientific, the generic name of water leaf, spear grass, grasses. Then, how is a quadrat used to estimate the population of it? They could ask you to describe how you can use quadrats to estimate the population of a quadrat. So you mark out the study area, that's the habitat, and peg the corners, toss the quadrat randomly within the marked area, count and record the number of target organism you found strictly inside the quadrat. Repeat this random tossing and counting at least 10 times in different spots. Calculate the average number of the species per row. Calculate the total population using the formula. So you can pause this slide to look at the detail and understand it more. So, final examination tips: why practical success formula. First, identify correctly. Make sure that you write only observable features. Use biological terms, draw neatly, label horizontally, follow the labeling guidelines, use ruler to draw your guidelines, complete practical tables accurately. You'll be provided with test instruction, follow the instruction to the latter. There will be a column for observation, write only what you can see, and then draw your inference based on the theoretical knowledge you know about food test. And then revise food test thoroughly, and then the ecological relationship between this specimen PQR, the thermometer, the meter rule, the quadrats, the ecological factor, their instrument for measuring ecological factor. Finally, this tutorial was painstakingly put together to encourage studying and proper preparation for the 2026 WAEC Biology Practical Examination. It is a postulated review of the specimen list asked provided by WAEC. You are to do your due diligence, go through the slides, understand the instructions, do your own personal review, shun malpractice. I do not support malpractice. I believe in due diligence. I believe in hard work. And if you can go through these slides and study it, passing is going to be effortlessly. Thank you for listening, for learning. This is Biology Clarified with Oluwatosin Ijewomen Ojo. I am also Tosin Lens. If you find this resource very useful, if you find this effort very beneficial, kindly subscribe to my YouTube, share this for other people to benefit. There are a lot of students out there who need this kind of review. Bye.